Schaeffer-Fulton stain. Microbiologist Eliot P. Schaeffer and MacDonald Fulton discovered endospore staining in 1913s, and this method is known as Schaeffer-Fulton stain. Here we use the Malachian green dye to identify the presence of endospore in bacteria. Schaeffer-Fulton stain. We stain the spores of bacteria. That is, spores are the resting phase of bacteria. The spore-related organisms are Clostridium tetani and Bacillus subtilis. We can do this Schaeffer-Fulton stain so that we can observe the presence of endospore in this bacteria. And spores have the tough outer covering called keratin. And in Schaeffer-Fulton method, a primary stain, Malachian green, is applied by steaming. While steaming, what will happen? Outermost layer, that is the keratin layer, will become thin. The spores will endorse into the cortex, and this cortex will consist of peptidoglycan layer. So this Malachian green will go and bind to the cortex or the peptidoglycan layer which is present in the cortex. So we will observe the spores in spores color will be a green color with the vegetative cell are counterstained with the safranin. So we can observe the vegetative cells in red color with a Malachian green or green colored spores here. So this is the procedure for Schaeffer-Fulton stain. So for the first step is the application of Malachian green to the smeared slide, and after that we will heat the slide. While heating, what will happen? This outermost layer of the endospore will become thinner, so easily the stain will enters into the endospore. Once it enters, we will use the declarator as the water. Just we will wash with water so that this endospore will resist declaration with the water. Next, we will add the counterstain, that is safranin. And after adding the safranin, again we will keep it for 1 minute, and we will wash the slide with water. After drying, we will observe the smear under oil immersion microscope. So we will get an observation like this. The Schaeffer-Fulton stain of Bacillus subtilis. Here you can see the green colored endospore with red colored bacilli. The red color is the stain of safranin and green color is the stain of malachite green. That means once the organism takes up the Malachian green, it resists decolorization with water. So the endospore will seen in green color with red colored bacilli here.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.